First, thanks very much to the organizers for inviting me to make this presentation on the origins and essentials of canine bioassay from the perspective of pharmaceutical research and development. And parenthetically, I'll state that I've been doing studies using dogs in both academic and industrial research since the mid-1970s. The agenda that I'm going to try to speak to is shown in this slide. I'll cover briefly the origins of dog usage in pharmaceutical R&D, talk specifically about who's currently using dogs and why. I want to spend a couple of minutes talking about the differences in bioassay versus hypothesis-based animal research study design, and then specifically address the role of regulation in the 20th century and the rise in the use of animals, and specifically animal bioassay for new pharmaceuticals. I'll end up talking briefly about who's currently making what types of decisions based on these dog bioassay data, and some summary and conclusions. So, covering the use of animals in science and specifically pharmaceutical R&D, I've identified 5 time periods starting with antiquity and ending with a 4th industrial revolution, which I've defined as 2001 going forward. I think you'll see in a minute why that last industrial revolution. But if you want further detail, I provided a couple of references here, which will go into greater detail of what was going on during each of these periods. But starting with antiquity, there is reference to the use of animals in ancient science. However, they are quite few. And I'll simply summarize by saying, in ancient times, it was much easier to use humans, whether they were conquered enemy, whether they were condemned criminals or slaves. They were readily available and I'll say easy to use. The first industrial revolution, roughly dating 1760 to 1840, is that period in Europe where there is a tremendous migrational shift from the farms into the growing cities. It's also a period characterized by the evolution of modern anatomy, surgery, and other disciplines, including physiology. And there was animal usage in this period, particularly in experimenting, demonstrating potential rudimentary surgical operations. I would point out during this period that, at least in the UK, the government consigned a proportion of condemned criminals. Once they had been executed by the government, their bodies could be publicly dissected by the Barbers Guild. So there was no shortage of human material during this period. You also may be aware that there was a lot of pilfering of fresh bodies from graves during this period by anatomists. The second period is 1860 to the beginning of the First World War. And this period is primarily characterized by the rise of modern chemistry, analytical chemistry, synthetic chemistry, medicinal chemistry. And the understanding here is that there was suddenly, for the first time in human history, a tremendous increase in numbers of new chemicals requiring therapeutic characterization to see whether they would be useful or not. You may be aware of Paul Ehrlich, who was involved during this period looking for a chemical cure for syphilis. And he and his colleague synthesized over 800 arsenical compounds before finding the first effective treatment for syphilis. Most of those compounds were not progressed because they were shown to be too toxic in rodents. The third period that we speak of, and this is the one I'll spend the most time with today, is the period after the Second World War to the end of the century. And you'll see more about that in subsequent slides. So here are the an analysis of data that was collected by the US Department of Agriculture under the auspices of the Animal Welfare Act, looking at the reporting of specific species used in animal research and teaching activities in the United States, 1973 to 2018, which was the last year the data were available when I did this analysis. On the left-hand side, you can see the statistics for dog usage. The values peak at approximately 200,000 dogs per year in the United States in the late 1970s, and then fall precipitously to approximately 60,000 dogs per year. On the right-hand side, just to put the dog data in context, I show you the data for all of the other species that are reportable under the Animal Welfare Act in the United States for the same period. And with one exception, they all show the same pattern as you saw with dogs, indicating that the fall off in dog use was not specific to dogs, but was specific to a much larger phenomena affecting use of animals, animal research in the United States, and as I'll show you in a minute, globally. The next slide addresses that single exception that I just referred to. Again, the blue line is the same dog data you've seen before. The orange line is the use of non-human primates, and this is predominantly cynomolgus rhesus macaques in the United States over the same period. And as you can see, although the absolute number is much less than dogs in the early years, there has been a steady, slow upward trend in the use of non-human primates over the same period that dog usage has so dramatically decreased. And I would surmise that much of that usage and increase has been due to the rise of biopharmaceuticals and the use of non-human primates in regulatory product testing for those products. You've undoubtedly heard that pigs are a substitute for dogs. So I've included here in the gray line USDA data for pigs. Prior to 2008, the USDA included pigs with all other farm animals, only breaking them out in that year. And I would just say that the use of pigs is following the same trend as the use of dogs. So, who's using these animals? The following data is again collected from the USDA database. Is for the year 2017, which was at the time the last year we had the full set of data. And collecting this data requires going back into the individual annual reports submitted by all of the reporting institutions to figure out which institutions are using what. In the left-hand slide here, we show the numbers of dogs used by non-research groups, government agencies, private companies, and research organizations and academic institutions. And we're identifying these sectors, organizations, by the names that they register with when they file their report. There are approximately 350 organizations in the United States reporting the use of dogs in 2017, and the majority of those organizations are private companies and research organizations, followed by academic institutions and, of course, very little government use of dogs. The academic institution use has to be considered carefully because there are an increasing number of academic organizations doing product development studies within their organizations, meaning they've built GLP capable facilities. But suffice it to say, in 2017, the majority of dogs being used in the United States were being used by organizations engaged in product development activities. In the upper right here, same data for non-human primates. And again, the pattern here is very similar as for dogs. I would say that, again, these data show only animals being used in research activities. They do not reflect animals being held but not used. So animals in breeding colonies are not represented here. But again, a similar pattern. Majority of these animals are being used by private research, industrial organizations, and again, presumably largely for product development. In the lower right, we see the same data for pigs, and here the pattern is slightly different: greater academic use, greater government use, and this probably reflects some of the shift away from the use of dogs towards pigs in basic research activities. Now, for contrast here, there is another country that has been very arduous in collecting annual statistics on animal use in scientific procedures, and that's the United Kingdom. The difference between the United Kingdom and the United States is in the United Kingdom, they count all vertebrate species, rats, dogs, fish, birds, amphibians, reptiles, you name it, and a couple of non-vertebrate species, octopus, for example, in their statistics. And they've been keeping these statistics since 1945. Now, the important observation here is total vertebrate animal use in the UK as reported species use in the US rose and reached a peak in the late 1970s and then fell off dramatically. There's a split here between the two blue lines. That's because in 1986, the UK changed its definition of what it counted as a procedure. The other point I want to make with this slide is the red line here at the bottom. That's if you break out the UK data so that you're looking at a similar basket of species as reported in the US. And the conclusions here are that the use of these species is very low. It's approximately 5% of the total numbers of vertebrate species being used, and it's effectively flat. So in conclusion, the most commonly used species in scientific and particularly pharmaceutical investigations have been and remain to this day humans. If you count the total numbers of humans used in a clinical development program, they will far exceed the numbers of animals in total used in all of research and development for that compound. And whilst dogs are mentioned since antiquity, the largest and systemic dog usage began after World War II and peaked within 30 years and has decreased ever since. The drivers for dog usage included and continue to include availability, tractability, relative size, and convenience. And specifically to the 20th century, the growth of animal and tissue bioassay as primary research tools in US and global academic and industrial research laboratories. The advent of systemic medicinal chemistry to produce new large numbers of compounds for evaluation. And as I'll discuss in a minute, new regulatory requirements. The primary driver for the decrease in dog usage in the latter 20th century was a scientific paradigm shift away from the traditional animal bioassay phenomenological endpoints to modern cellular and molecular biology and molecular endpoints. Currently, the use of dogs in biomedical research is predominantly for purposes of regulated product development and satisfaction of regulatory requirements. And this would include pharmaceutical, industrial, and for agricultural chemicals. Now, as I said, I wanted to just spend a minute to refresh you with the subtle but not very significant differences between animal research that is hypothesis-based versus bioassay-based. In hypothesis testing, the study is directed at a prospectively identified molecular target or targets or a pathway. There is an anticipated pharmacologic, pharmacokinetic, etc. response. Hence, there are specific experimental endpoints driven by the hypothesis. It's reductionist. It's only applicable to previously identified mechanisms. It has applications in discovery and development for screening, for identification and mechanism of action, for proof of principle. These studies are often characterized by the sorts of summary characteristics like an effective dose 50%, an inhibitory dose 50%, a response threshold, a maximum response. And cross-species translation via mechanism and pathway analysis is entirely feasible with hypothesis testing. Contrast that with bioassay testing, where there is no prospective experimental hypothesis. There are no anticipated responses, only routine endpoints. Could be blood pressure, heart rate, or clinical observations, hematology, clinical biochemistry, histopath, etc. The responses are integrative. What one will see in these studies is a summation of direct actions of the test article in combination with any reflex pharmacological, physiological reactions. Applications are again in screening, therapeutic target identification, hazard identification, and safety assessment. The sorts of summary statistics that you'll see with these studies are different. They'll include items like a lethal dose 50%, a maximum tolerated dose, lowest observable effect level, no observable effect level. And cross-species translation is dependent upon establishment of chemical or mechanistic linkages. These are not trivial studies to conduct, and I just summarize in this slide the sorts of, excuse me, items that the study director and the study team have to be on top of, in control of, in order to complete the bioassay. And these include the quality of the test article, preventing cross-contamination, control of husbandry issues. Bedding can be a big issue. Human interactions, the degree of training and experience with specific procedures, environmental conditions, air quality, temperature, lighting, and often underappreciated, the quality and composition of the diet, contaminants in the diet, daily intake. All of these form this, if you will, black box in which one is doing the experiment, and inside the black box is the experimental animal. I want to change gears here and speak to the role of regulation in the 20th century, driving the use of animals, and particularly the use of dogs, in pharmaceutical product development. The first was the 1937 Pure Food and Drug Act, followed by the 1962 Kefauver Amendment. The result of these two events was the requirement for routine animal safety testing of new drugs as a prerequisite for US clinical trials and marketing approvals. In 1962, Marshall Bioresources initiates raising beagle dogs for research. And they do this in response to the changes imposed by the US FDA for non-clinical testing of drug candidates. This was codified in the 1982 FDA Red Book 1, Toxicological Principles for the Safety Assessment of Food Ingredients. And although its title states food ingredients, the Red Book is used for all of the products that the FDA regulates. And it codifies the agency's two species, one rodent, one non-rodent paradigm for non-clinical safety evaluation. It also specifically states that dogs are the preferred non-rodent species. Now, in 1982, if you were a drug developer, you would have a non-clinical study package designed to meet the requirements of the FDA, and you potentially had separate packages to meet the requirements of the European Union and Japan. And this, as you can appreciate, led to a lot of duplication of studies, unnecessary use of animals. The result was the formation of the International Conference on Harmonization of Technical Requirements for Registration of Pharmaceuticals for Human Use, so-called ICH. And there are three guidances from ICH that speak specifically to the use of non-rodent species in general and dogs in particular. And they are the M3 guidance for non-clinical studies, conduct of human trials, and then the two safety pharmacology guidances. ICH 7B is the guidance regarding drug-induced delayed ventricular repolarization, or drug-induced long QT syndrome. Speaking to the duration of the repeat dose toxicity bioassays to support the conduct of human clinical trials, this is from ICH M3R2. Up to 2 weeks clinical study duration, you'll need 2 weeks in rodent and non-rodent species. Between 2 weeks and 6 months, clinical trial duration, it's day for day. And greater than 6 months duration, you're going to need a 6-month rodent and a 9-month non-rodent study. Now, we could spend a lot of time talking about why 6 months in rodents and 9 months in non-rodents. But I would just suggest to you to remember that for most pharmaceuticals, the regulatory agencies are going to get a 2-year carcinogenicity bioassay in 1 or even 2 rodent species. Hence, I suggest to you why they were willing to compromise on the duration of the chronic tox study. The goals of the non-rodent or canine bioassay from ICH-M3R2 is primarily to identify and characterize toxic effects with respect to target organs, dose dependence, relationship to test article exposure, and when appropriate, to identify the potential reversibility of an adverse effect. Again, I present this to show you that there is a considerable amount of thinking that goes into the design of these bioassays even today. And you'll see this in terms of the justifications that have to be provided by the study director for the design of the study, including justification for the species. Particularly here is demonstrating the capability of the selected species to exhibit a primary pharmacological response to the test article. Males and females are usually used because even if the pharmaceutical is designed only for males or only for female therapeutic condition, both genders are likely to be exposed in the supply chain. Young adults at necropsy are preferred in order to avoid confusion of test article changes with juvenile characteristics. There will be a justification for the dose, the formulation, the route of administration, and as you saw before, the duration. The route of administration will be the best approximation of the proposed human usage. The high dose will be either a maximum tolerated dose, a maximum feasible dose, limited by the route of administration, a limit dose, a gram per kilo per day in general, or sometimes a 50x multiple of the anticipated human dose exposure. Now, the key word here is anticipated, because at this point, the drug developer won't have any information as to what that dose is, and hence can become limited if one chooses this route. There'll be further justifications for monitoring, sample collection, method of euthanasia, postmortem collections, and I would say parenthetically, virtually all of these studies are conducted to the good laboratory practice standard. Now, one of the reasons I mention this is I'm sure that everybody attending is aware of the problem with reproducibility of scientific study results, both animal and non-animal. And the point that I would make to you is the one thing you haven't heard are any issues around the reproducibility of these animal bioassays conducted in support of product development activities. I can tell you from personal experience that there have been attempts to test the reproducibility of these findings, and in all of the cases I'm aware of, there was reproducibility across organizations and across continents. I'll leave it at that. How are the bioassay data applied? Well, as specified in ICH M3R2, a primary application is to identify non-clinical target organs of potential safety concern in humans. Also to estimate an initial safe starting dose and dose range for the human clinical trials. That's a separate guidance document if you're interested in the details. The bioassay studies also identify clinical monitoring for potential adverse effects. This would be monitoring other than the ordinary stuff. And the bioassays provide bases for identification, characterization of safety mechanisms leading to human translation, design of additional non-clinical studies to evaluate, mitigate human risk, to identify antidote and rescue strategies if needed, and by the discovery organization for design selection of backup and second generation compounds. Looking more broadly, the data does contribute to ecological and environmental analyses. This is this issue of when pharmaceuticals get into the environment, either through sanitary waste systems or other means. And finally, and perhaps not generally appreciated, the bioassay data, particularly using dogs, is critical to provide dosing and safety data for veterinary use of human pharmaceuticals in the absence of an approved veterinary product. And this often occurs with anti-cancer strategies. So in summary, Canine Bioassay has made immense contributions to science and human and veterinary medicine over time. The canine data is suggestive, not predictive, of cross-species translation. And the canine data continue to be used by multiple stakeholders for decision-making in pharmaceutical R&D and human and veterinary medicine. I'll conclude by reminding the audience that if you were working in this area 30 some odd years ago, you would be aware of a desire to find that perfect animal model that predicted both efficacy and safety for all new compounds that you were taking into humans. And the reality is there is no perfect animal model. Hence, as a result of my career, I'm now a proponent of George Box's wisdom. All models are wrong, but some are useful. And with that, thank you very much.